genetic epidemiologist, my passion is understanding heterogeneity in different populations. It stems from my own experience as an individual of Mexican descent. And in epi, we're very good about sort of lumping things into boxes, right? And not looking at within group variation very often. And so in my studies of diabetes and cardiovascular disease and obesity, it occurred to me that we were classifying Dominicans and Mexicans and Cubans and all of these other background groups of Hispanic Latinos into one category and looking at associations of disease with all of them together, right, as one. And so that one group in particular might be driving the association. Now, why do we do this?
Small groups.
Small groups. It's so hard, right? So they're underrepresented sometimes in research, so which is one reason why we categorize all into one group. Sometimes because we do have such small numbers, we remove them from the analysis altogether. And so I started as an applied epidemiologist looking at incidence and prevalence of HPV and cervical cancer in Latina immigrants, and then I had an opportunity to pursue a postdoc, and at the time had how old was he? 2-year-old? Yes, 2 years old. 3 years old. 3-year-old son who was going to have to go to daycare somewhere, and it was either the cardiovascular disease T32 at Harvard, where I was going to pay $3,000 for daycare, and I was going to make $4,000 as a postdoc, or a postdoc in statistical genetics at UAB, which I ended up doing. And so I switched gears a little bit, and I have joined the field of genetic epidemiology. I'm now an investigator in the Top Med Project, which is the Transomics for Precision Medicine initiative that is NHLBI funded, and so very integrated into precision medicine now. And with my applied head on, thinking how that fits into precision public health, right? Which are sort of two different things. When you think of precision public health, you think of the technologies that you're going to use to make interventions more precise, right? You might think about surveillance and informatics, or you might think about also targeted interventions very generally. And so that's how I arrived at what I'm now sort of piecing together is how to integrate genomics and cardiometabolic disease and implementation science. So I actually just completed a 2-year fellowship in implementation science at UCSF with Kirsten Bivens Domingo and others who are very focused on trying to better understand how you take evidence-based findings and implement them and disseminate them in the community and hospitals and sort of more generally. So I have no disclosures. In that time, you know, as you're training as a fellow, you have all of these exercises that you're engaged in, and they want you to take your research and see how you can create an implementation plan. And so as a genetic epidemiologist, I thought, well, is there anything in genomics that's ready for implementation, right? Maybe on the pharmacological end, but I don't do pharmacogenomics. So it was hard for me to find an example or a project that was ready for implementation and that I could use to sort of guide my studies in implementation science. And I thought, well, why is that? So let me think about why that is. And so I went back to, you always go back to the basics, right? So what's the definition of implementation science? And it's the study of methods to promote the adoption and integration of evidence-based practices, interventions and policies into routine healthcare and public health settings. Now, one could argue that evidence-based findings are limited in any field, right? Depending on the field you're in, they may be very limited to begin with. So it's not particular to genomics. Okay, so for genomics, I went back and I thought, okay, well, where does my work lie? And so I don't have this slide, but many of you may have seen the like T1 to T4 graphic, right? Where you have sort of the basic science and you take it through the different stages of translation to where you're now able to translate the evidence that you have from your research into clinical settings or population settings or communities or what have you. So I'm really sort of T1, right, in genomics. I have all of this data now that I'm working with, so I do all of these omics, maybe not proteomics so much, but epigenomics, transcriptomics, GWAS, metabolomics. The top med project is whole genome sequence, so we have 150,000 or more whole genomes that we've collected so far, and we have 3 billion data points per genome to study. So that's sort of what I'm working with, and then there's all of this other work that happens to get to what is causing disease. So this is a graphic that I found in Cancer Epidemiology Biomarkers by Revick and others. And I thought it really gave a great comprehensive look at what precision health is. And so we start at the beginning asking, what do we know about the disease? So what's the fundamental research about behavior and biology and biomarkers and so forth? That gives us a sense of which individuals are at risk, or we ask the question, which individuals are at risk. Through community-engaged research, we can ask where are the communities of needs. Things like geospatial research can help us answer that question. Or we can come down and ask what prevention strategies exist. In whom is prevention effective? How do we get the intervention to be relevant to the individual or community? There's all these questions in all of these stages of research that have to happen before we can get to the implementation stage. Or even arrive at creating precision health, right? Precision medicine is something altogether, but precision health, I think, is a lot more complex than that. So how do we arrive at our understanding of genomics in diverse populations? Okay, so back to my question. So getting back to the issue of these heterogeneous populations, right? My interest in disease, which is cardiometabolic diseases, and how that all pieces together. So the study of diverse populations. So as of 2016, diversity in genomic studies has increased. We've come some way compared to where we were, I don't know, at the advent of GWAS, for example. But even so, still underrepresented are Hispanic Latinos, Pacific Islanders, Arab and Middle Eastern individuals, and Native Americans. We have very, very, very small sample sizes, and even for Asians. So I think Asians are quite diverse in terms of heterogeneity and the groups that are represented within that single category. And so those individuals are underrepresented if you think about how many groups are comprised within that one group. So we've made some headway, but not enough, I would say. And why is that important? So in genomics, those of you who are familiar with some of the genetic studies that have been done to date may have heard about GWAS, right? Which is genome-wide association studies. So GWAS look at common SNPs, or common single nucleotide polymorphisms. So a single base pair change, right? And its association to disease. Now, part of the complication with GWAS is that what those studies were using sort of as a reference had very little diversity to begin with, right? And so what inferences you could make about the generalizability of those results was very limited. We've come a little bit farther away with respect to those reference panels, but I think we're still not quite at the point where we can make sufficient or accurate references, or inferences rather, about the associations of disease using the reference panels that exist. And we're making some progress with that. So now, for example, with the Hispanic Community Health Study, or the study of Latinos, which has 16,000 Hispanic Latinos that were recruited from 4 different sites in the US, has 6 different background groups of Latinos. There was actually work to develop a chip that would include the alleles that are more frequent in Hispanic Latinos, and so that was one project that was specific to that population, which has helped advance that work. But there's a missing genetic link that GWAS did not cover, and that is rare variants, right? So this is where the diversity of populations is very important. So rare genetic variants are those that occur in less than 5% of the world's population, but comprise the bulk of genetic variants. May be disproportionately important, both in determining the risk of disease for a person, but also in getting complex disease understanding and predicting the response, for example, to a particular drug. So a lot of this work has been shown in the study of warfarin, right? I don't know if you're familiar with that. So those with African ancestry have a different response to warfarin, which is the blood-thinning drug, compared to individuals of non-African ancestry. For those of you that are interested, Nita Limdi is an investigator at UAB who does a lot of this work and did a lot of the very seminal work in this area as well. So, rare variants tend to be population specific. So it's very important that when we're looking at rare variation, that we're actually looking at rare variation in a very diverse sample of individuals, right? Which comes with its own complexities. So if they play a key part in disease, the lack of diversity in genetic studies will be severely skewing our understanding of which genetic variants are important. So, Charles Martini, anyone familiar with him? Yes.
Yeah, my first job in public health was at Loyola Medical Center in Chicago, and he was on faculty there.
So he leads a big center now in DC for the study of diversity, largely African diversity, but has written a lot of the seminal papers in genomics and heterogeneity. So he's one to look up if you don't already know about him, and he's the current Center Director for Research on Genomics and Global Health. So he's famous for saying that our genomes are a history book that unbiasedly captured human experiences over thousands of years. And we can use this history book to learn about history and health. It helps us better understand medical outcomes and medical treatments, right? Because medicine in itself is an inexact science. And so there's a lot of guesswork that's inherent in medicine and the practices that are used to increase efficacy of the interventions. Genetics is one important contributor to this complexity. For example, distinct genetic variants may cause conditions that, like I said, make people respond differently to treatments or share a similar set of symptoms. But it also helps us identify groups at risk for certain diseases, which I'll give you an example of for type 2 diabetes in my own work. So let's take type 2 diabetes, for example. It accounts for 90 to 95% of what's called diabetes. It includes type 1 diabetes as well. The genetic variants come from over 100 genes. They contribute to risk with some very small additive effects, right, across the 100 genes. We've seen this largely in populations of European ancestry. In studies of ancestral groups, there are differences in the variant prevalence. Some variants are found exclusively or predominantly in specific groups, and some have strong genetic evidence of T2D susceptibility. So the Pima Indian example is one that is classic in type 2 diabetes research. So they have the highest reported prevalence of diabetes of any population in the world. I mean, across the board. This population provides researchers with the opportunity to study diabetes in a population with very limited genetic and environmental exposures. This is one example of where I forgot to bring forward and bring back. So sorry, sorry that that's cut off. But that's the abstract if anyone's interested in reading the original paper on that. So prevalence is much higher in these ethnic groups than in others, even if they're in the same countries. And these observations, together with the evidence of the important genetic susceptibility to type 2 diabetes, indicate that type 2 diabetes results from an interaction between genetic predisposition and lifestyles associated with unfavorable environments. So there have been studies of Pima Indians in Mexico and also in the US, right? Adult Pima Indian and non-Pima Indian population in the Sierra Mountains of Mexico were examined. So they were given an oral glucose tolerance test, OGTT, and assessments for obesity and physical activity and then other things. And the results were compared with those Pima Indians from Arizona. So a comparison of the same population in two different geographic locations. Both Pima Indians were typed for DNA polymorphisms to establish genetic similarity. And what they found was that the age-adjusted prevalence with 95% confidence intervals of diabetes in non-Pima Indians, Mexicans, and Mexican Pima Indians, and US Pima Indians. So non-Pima Mexicans, Mexican Pimas, and US Pimas. And you see that the prevalence of type 2 diabetes in the US Pimas was astronomical. Physical activity. So this was 2.5 fold higher for men and 7-fold higher for women among the Mexican Pima. And obesity was also 10-fold higher for men and 3-fold higher for women in the US Pima. So the much lower prevalence of type 2 diabetes in the Pima Indians in Mexico than in the US indicates that there might be some environmental contributor, right? In addition to the genetic predisposition to type 2 diabetes. Because we already know they're genetically predisposed. There's some environmental factor. So this is kind of getting at what Dr. Lee was talking about earlier with respect to, the argument that race is race, right? So there's a genetic component to their race ethnicity that is existing. That's sort of like their constant, right? The genetic predisposition to type 2 diabetes that's attributed to being Mexican or Pima Indian rather is constant. But there's this other sort of factor that's contributing to risk of disease in this case, and that may be the environment. So then we get to soul. So I've been very interested, as I said, in the contributions of this heterogeneity, right, that exists within Hispanic Latinos and sort of how we group them, the idea of disaggregating them. And this is an analysis of Hispanic Latinos compared to Europeans, Africans, and Native Americans. And then this is an analysis of ancestry from Mexico, Ecuador, Colombia, Puerto Rico, and the Dominican Republic. And the gradation of color sort of shows you the variability that exists within each group. So this is taken from the Human Condition, Chapter 8, Genome-wide Patterns of Population Structure. And basically what it tells you is that Hispanic Latinos, because they are sort of grouped together, but even when you look at them individually, there's a lot of heterogeneity there compared to other populations. And in fact, there was a report that was written by the Harvard Institute Research Working Group. So this was published in 2002, and it looked at the diversity around the world, right? With the darker green being more ethnically diverse, and the sort of orangey brown being more ethnically homogeneous. And you actually see that Rwanda and Liberia are the two most ethnically diverse countries in the world. And places like Mexico, for example, where one would expect, we are lumping all of this data together, so perhaps there isn't that much diversity, but in fact, they're pretty green, right, on that scale. So I'm going to go back to this one more time. There's a paper by Esteban Burchard and Carlos Bustamante who looked at the genetic heterogeneity in Mexico, and there are actually points of heterogeneity differences across the map of Mexico. This was with respect to prevalence of asthma. And so that paper is out there, and I can share that reference if necessary. All right, so bringing this all back and sort of, by now I've convinced you that it's important to look at diversity in studies, and especially genomic studies. I want to present you the case of LDL. And I'm not a clinician, I'm an epidemiologist. I think the only epidemiologist who's speaking at the workshop today. And so when I think about things like LDL and HDL, I have to sort of give myself clues as to which one is which, and I always thought of LDL as sort of like the low down dirty lipoprotein. The bad one, not the good one. So the case of LDL. Okay, so here we have a low plasma level of LDL is associated with reduced risk of coronary heart disease. So if you have high LDL, you're more likely to have coronary heart disease. So